Right. So welcome everyone to the third edition of the DMS Pro workshop. And as you can see here today, we are going to talk about the particulate turnover of methylated sulfur compounds, trying to bridge microscale processes and measurable grazing rates and particulate turnover rates. So this session has is a bit different compared to previous sessions, which compare different methodological approaches mostly. And we have today two fantastic speakers who are Katherina Petrou and Steve Archer. So before we go to the talks, let me give this brief introduction, which I already gave a couple of times before, but let's repeat it because some participants have not attended previous workshops. So DMS Pro is a SCOR working group which has the objective of developing resources for the study of methylated sulfur compound cycling processes, so focusing on rate measurements. And here you can see the 21 members of this group from many different countries. And what were the motivations to create this group? Well, the idea was to re-energize the study of these compounds in the ocean from different points of view, and to this end we devised a number of activities and deliver and products we want to deliver, which include creating standard operating procedures, so reaching kind of community agreement or at least community discussion about the different methodologies, what they measure and what they don't. And in fact, this discussion today should be very informative for creating this standard operating procedures document, which we plan to write in the following months or year, more realistically. Then we are also going to create a database of cycling rate measurements, which should be very useful for comparing each own's measurements and also for model development and validation. And we are planning to write some articles giving perspectives and describing the database and giving guidelines for modelers and so on. All of this like open access to make it as widely used as possible. Great. So here you can see this classical schematic of methylated sulfur compound cycling processes, which is quite comprehensive. It includes lots of processes in the particulate and dissolved phase, some of which we have covered in previous workshops, like the consumption of dissolved substrates and the synthesis of DMSP. And one motivation to create DMS Pro is that, in fact, we after like more than 15 years after this schematic was created, we know that it needs several updates. And also another motivation was that during the last decade, there has been a decline in the number of studies measuring cycling rates, which is unfortunate because this matches the increase in the molecular techniques. And we all agree that combining molecular techniques and rate measurements makes everything more powerful. So we want to ideally revert this trend. And with this, we arrive to the two speakers today, who are Katherina Petrou from the University of Technology, Sydney, and Steve Archer from the Bigelow Laboratory for Ocean Science. And as I explained before, today's session, we are trying to bridge somehow microscale interactions, which are the ultimate driving processes of these particulate turnover rates, and the turnover rates we can actually measure when we take a matter sample and incubate it. And we hope that each of the talks will shed light on the other scale or type of approach. How does the workshop function? So after this for a brief introduction, we have the two talks. We have ideally time for a few quick questions, so we don't forget what we wanted to ask our speakers. After that, we create two breakout groups. And now I encourage everyone to participate in these breakout groups and not leave the the workshop after the talks. And in this, well, we are separating we are splitting in two, depending on whether we are experienced users of the methods or have some experience like making this any kind of measurements, or we are new in this field of study. And well, the whole point of this way of this splitting of the group is that everyone feels comfortable discussing together and to maximize participation. So please don't be shy and try to to participate in the in the discussion. Everyone is welcome, and all points of view are very welcome. After that, we gather again to have a collective discussion, which will last 20-30 minutes or more, depending on how is it going. So, having said this, the floor is yours, Katherina, and I will stop sharing my screen.
Thanks, Matti.
Welcome.
All right. I need to work out which screen I'm sharing now. Can you all hear me all right?
Yes.
Yep, excellent. So I'm going to preface this talk with the fact that I'm home alone with my 3-year-old who loves to wake up at about this time. So fingers crossed I get through this talk without any tears. His, not mine. So, all right, I'm actually going to be jumping my slides, talking a little bit more of the dissolved component and how that's cycled in the microscale, but that should segue nicely into Steve's talk following. So thanks for having me. This is really exciting. And we're going to look at the ocean at the microscale. So when we look at the ocean at the microscale, we're really talking about the marine microbial world. And in that context, distance, diffusion, and dependence are going to be really key aspects to understanding sulfur transfer and metabolism. So here we have our lovely DMSP producer or compound producer, and it's in this 3D ocean environment. And what we have to remember is that for microbes or cells, distances are really large. So unless there's a really big bloom, there's got to be some sort of way to find the compound of interest, and motility can be really important. Diffusion is a function of cell size and concentration. So again, distance, diffusion, and dependence or concentration is going to make a difference as to who finds what. What's really interesting is that chemicals produced by one organism can be essential, like B12 by bacteria is essential for phytoplankton, or simply desired. And sometimes we don't really know what the main driver is for that attraction. But what we do know is that methylated compounds underpin many really important ecological relationships in the ocean. So what do I mean by that? Well, when I'm talking about ecological relationships, I'm referring to sulfur producers, sulfur metabolizers, those that can use it as a trigger or cue, chemical cue for waking up from dormancy, and those that can take it up or be a reservoir or sink, yet not necessarily known why. So these are the 4 areas I'm going to briefly go through in my talk today. So sulfur as a chemoattractant is a really cool area of research, and one that I've sort of been a little bit involved with, but also on the periphery of. And it really started back with a colleague of mine, Justin Seymour, and Raphael, who I was hoping would be here today, with some really cool work in 2010 where they looked at the chemotactic response of diffusing patches of DMSP with different microorganisms. So they had Pseudo-Ultramonas, Micromonas, and Oxyrus marina, and they could see using this a much more sophisticated flow cell than this, I'm sure. They were able to have these patches and then look at the concentration of those cells as they were drawn to those patches. So really looking at that chemotactic response. Justin continued a lot of this work back in Australia, and one of his PhD students, Jessica Tout, I worked with them on a project that she went to the reef and wanted to look at chemotaxis in coral-associated bacteria. So she went out and collected some seawater that was close to coral to get that, and did some very simple chemotaxis assays just using a syringe filled with DMSP inside a jar, put the water in and looked at the cell density and molecular 16S of what came in that syringe. But with the cell density, she found that DMSP created a very high chemotactic index. So these bacteria really did seem to respond to relatively high DMSP concentrations. So she took that idea and went into the environment in situ, into the reef, and she used a very early version of this lovely ISCA, the in situ chemotaxis assay. And I'm really glad JB's here with us here today, because this has developed a lot over the last decade, and JB's been at the forefront. So he can answer all your questions relating to that later. But what Jess Tout did is she placed these out on the reef at open ocean site at the reef crest with the croperids known to produce a lot of DMSP in a reef and then in some sand. And she found very strong chemo attraction with these, or chemotaxis with these bacteria in association with the corals. So the Acropora aspera at the reef crest. And it was for a bunch of compounds, but within that was DMSP as well. And this work, as I said, has developed and continued substantially in the last 9 years or so. Some recent work by JB and colleagues actually started looking at how DMSP can enhance chemotactic responses. So they showed that there's chemotactic responses to these polysaccharides exuded by macroalgae, but that by having DMSP present, it facilitated or increased that response. So some really cool work is coming out that's pushing the boundary and pushing that further. Some even more recent work, which was by Guel-Bouillon, I don't know how to pronounce the name, but Raphael's part of that group too. And is this microcapillary system that they used, filling these microcapillaries with DMSP and a control. You can see here, DMSP versus control, and finding chemotaxis in 3 marine herbivore protists. And the idea behind this research was really to look at this idea of attraction versus deterrence with acrylate, DMS, and DMSP, and that grazing response. So again, looking at turnover in the ocean. But they found very strong chemotaxis in all 3 taxa towards DMSP. Excuse me. If we zoom out for a second, that's sort of really taking a microscale approach, but to try and scale up, we can use molecular approaches and look at microbial associations. So a PhD student of mine and Justin's, James O'Brien, did some really interesting work taking genome data from around Australia, from these national reference sites that we have around the continent. They're sampled monthly for genomics, and he used 3 years worth of data, and it's also depth resolved. And he took the relative abundance of the Rosiobacter group, which are known DMSP catabolizers. He also then took the relative abundance of DMSP producing phytoplankton, building on the work of Erin, who's, I believe, with us today. Erin, who's in this work, and Naomi's looking at those prominent producers versus less prominent producers, but really trying to hone in on the DMSP producing phytoplankton. And what he found was some really strong positive Rosiobacter DMSP producer interactions. So we have the Amylobacter with Micromonas, we have Rhodobacteriaceae, and the strongest one was in B up there was the Sulfidobacter with Phaeocystis and Thalassiocyra. So there's some really interesting patterns at a larger continental scale can really be looked at, and it's suspected that DMSP is the underlying, or at least sulfur compounds are the underlying attraction between the two. We did some similar work down in Antarctica, where we had the opportunity to sample along a glacier out to the open ocean, so along this gradient. Amongst environmental gradients, we also had a DMSP gradient, so we measured DMSP dissolved and DLA. We also see an increase in chlorophyll and, in particular, the hex fucoxanthin, the marker for Phaeocystis, as we headed from inner to outer glacier to the open ocean. And using our 16S and 18S, we were able to get distinct communities from the inner to the outer reef. So inner being reef, inner to the outer glacier, inner being the blue and outer ocean being the brown in both the bacterial and phytoplankton components. And looking at that as a network, it's a little bit fuzzy, I apologize. But we had this very tight cluster around dissolved DMSP and DMSP lyase. And of particular note was the hexfucoxanthin marker for Phaeocystis, but some of these really large diatoms were also associated strongly. What's links to James's work was that we found Sulfidobacter, Unioloctonella, Octodecabacter, and Amylobacter all associated with that group. And they're part of the Rhodobacteriaceae again. So those phytoplankton-associated bacteria are very strongly linked to the high DMSP. These were unrelated from the inner reef groups that we had. So some interesting work coming out there. So we found strong evidence, there is strong evidence for chemotaxis towards DMSP, but we need to remember that the ocean's a really big place for these organisms. So density and proximity are going to be important, and motility to be able to get to the source. But we know that methylated compounds do govern a lot of interdomain associations in the ocean, which are important for sulfur cycling. So the next study I wanted to bring up was the use of sulfur as a chemical cue. And this was my introduction to sulfur, actually, as of my first postdoc fellowship with Rafael. I got an opportunity to go and work with Rafael in Barcelona. And while I was there learning about sulfur, we got to work with Esther Garces and her team on these very cool parasitoids. This is Parvuliciferous cinerae, which is a parasitoid that can infect the toxic dinoflagellate Alexandrium. And it does this readily and terminates blooms, so it's quite an important ecological organism. The infection cycle starts with a Parvulicifera infecting a cell, dinoflagellate, and then dividing and growing and hollowing out the dinoflagellate until it sheds it and becomes this what's called a sporangium, this ball of cells waiting for a release signal. What they observed was that the sporangium would only release the new Parvulicifera in the presence of Alexandrium. So we jumped on that idea and thought, this is really cool. Let's have a look at that chemical cue, or if it is a chemical cue. So we took these sporangia and we had a control, obviously, but over time we added A. minutum cells and measured their release over time. So the number of activated sporangia. And we saw that it did it with both the Alexandrium present and the exudate. So we measured the compounds in the exudate and added those and found that acrylate and DMSP had no effect, but DMS at the same concentration had the same effect. So we'd found our chemical signal. And we tested that with different concentrations and found there was a threshold, a minimum threshold for the release. So we were able to see that DMS caused the release of the opening of these Parvuliciferous sporangia. So it was this wake-up signal and that it was concentration dependent. So we'd found this trigger for dormant to active stage. And it's a really effective bloom termination strategy, but also survival strategy. We did do a follow-up experiment with chemotaxis, and there was no chemotactic response, interestingly enough. So it is just this, is there a bloom? Are we ready to infect? And they release. So that was some really cool work there. And the last thing I was going to share with you was the rest of the phytoplankton. So assimilation and osmotropic uptake by other phytoplankton. And this work started with Villa Costa et al., who did some work using 35S, so things we talked about in the first workshop. And it was very nifty because they put that in and actually found that the picoplankton, so the cyanobacterium phytoplankton, picoeukaryotes, actually took up a lot of the sulfur in sulfur, it's 35S DMSP. And that was really interesting. And they also did autoradiography, microautoradiography, to look at the larger fractions and found that also diatoms seem to contribute to this uptake of sulfur as well. I really liked this work and just started exploring that in my lab. So we started looking at using Thalassiocyra visflogii, a large centric diatom, or large relatively, and looking at uptake rates. And sure enough, it takes up and withholds or retains DMSP to very high concentrations. And we looked at concentration-dependent uptake, but also salinity responses. So we did multiple different salinities to see how it affected uptake rates. And then loading cells, we also looked at how it might work with quenching ROS, so reactive oxygen species in the cell, and start probing these questions using this model diatom loaded with DMSP. And that's some interesting work that we're still pursuing at the moment, and it's kind of cool fun. In a similar vein, looking at assimilation in natural environments, a PhD student of mine, Eva Fernandez, looked at two sites on the Great Barrier Reef. She was on a voyage and she did an inner reef and an outer reef site where she was able to look at DMSP uptake in the microbial community. The inner reef site was hypothesized to be high DMSP because of the coral producing, and the outer reef site more oligotrophic. So she took water from both sites and incubated them in the presence and absence, as well as fixed, of DMSP enrichment, and then fractionated the community to look at the particulate or uptake of that DMSP. And what she found was that at the outer reef site, the oligotrophic site, DMSP uptake seemed to be dominated by the smaller size fraction, whereas the larger fractions at the inner reef seemed to be responsible for this uptake. And we did some molecular work after that to look at who those taxa were. We conducted a similar study in Antarctica, and it showed a very similar thing. This large fraction, particularly at the very large fraction of the large diatoms, seemed to take up a lot of the available DMSP, and that was in excess because there was already a high concentration of DMSP in the water sample. With these studies, we actually added 13C DMSP, so the stable isotope, and we ran the treatment as well, and to do some stable isotope probing on the community and really look at which group was taking up the DMSP, which species. Unfortunately, that didn't turn out so well, so I'm hoping we can redress that at some point. But the uptake results were very similar between DMSP and 13C DMSP, so the bars look the same. So all that really highlights is that DMSP is shared across the microbial community and phytoplankton community, including these non-producers. The ecological importance of multiple sources and sinks in the sulfur cycle is yet to be fully resolved, and that the physiological role of DMSP in these sinks is relatively unknown. So we're still exploring that. So on that note, DMS can act as a trigger for these awesome little parasites or parasitoids. DMSP can work as a really strong chemoattractant in the ocean, but also form strong microbial associations. And it can be also taken up for some purpose by these potentially temporary reservoirs or sinks in our ocean. So on that note, I will let you zoom out a little and hand over to Steve for some more higher trophic level grazing discussion. Thank you. And Darwin didn't wake up. Yes.
Yes. Thanks, Katherina, for the fantastic talk. And well, for giving this talk at this time of the day in Sydney.
Yeah, it's Friday. Happy Friday, everyone.
Well, that's true. So we have time for a couple of quick questions, I guess. So please go ahead if if someone wants to ask something now. You can raise your hands or just directly jump in and ask Katherina. Yeah, Jacqueline.
Hi, yeah, thank you for a very interesting talk. Thanks for that. I was just wondering what method you used for the DMSP uptake measurements.
So for the cells, okay, so we did dissolved, so the dissolved fraction for the amount added to see what was removed, and then the particulate. So we did using GC with direct injection. So we simply did the sodium hydroxides on the filter. So we filtered the sample and did the particulate and dissolved fractions.
Okay, so you didn't use stable isotopes for incorporation. So it's basically referring, you're not entirely sure.
Correct.
Whether what you lost in your dissolved fraction is actually ending up there, but you refer it from a mass balance.
Yes, correct.
Okay. All right. Yeah. Okay. Thanks.
That's all right. I would love to use stable isotopes. We tried with one set and it just didn't seem to work for us.
Yeah. Nice.
I guess I have a question. Nice talk,
Thanks.
Katherina. The question is, in the chemoattractant work, does anyone look at how how the past response at a concentration affects the future response? Do the microbes become used to a level and they no longer see it? You know, like if you go to a room and you smell something and you're there for a while and you don't smell it anymore, is that something
Great
people think about?
question. That's a great question. I don't believe so, but I can also open the floor. JB, I don't know if you have any idea on that one, since you've done a lot of
Yeah.
chemotaxis work.
Hey, Dave.
Hey, JB.
It's a very good intuition. If you have too much of this molecule, the receptors will be saturated, and the microbes will not be able to sense a gradient. But you need extreme concentrations, much higher than what's typically present in the environment.
There was a Jonathan, I think Jonathan had his hand up, but I don't know where he's run away to. He might have fallen. Oh, there you are.
Thank you. Yeah, sorry. Yeah, just a very quick question. So when you talk about assimilation, do you mean import and use of DMSP as like for its anti-stress properties, or do you mean actually it's used as a nutrient?
That's exactly what we're trying to ascertain. So we've got the uptake. We know it's in the cell, and we did get some evidence for it reducing ROS in the cell. So the ones that were loaded with DMSP did have reduced ROS during growth, different growth phases than the ones that didn't, the cells that didn't. But we haven't made that official link yet, and I would love to do more work. You've just jumped over here. Love to do more work to really tease that out, both with ROS, but also salinity stress and various others. And I've actually got a student starting to look at that in particular, and also the localization of it in the cell to really try and nail down where it is and what it's doing over time. Yeah.
Marty, you're muted.
Sorry. I was saying thanks, Katherina, and again.
Thank you.
And we can move on to Steve's talk. Thanks for telling me.
Okay, can everyone see that? And can you hear me? Great. Okay, well, I've already got ahead of myself, which isn't too surprising, but I'll go back one. And yeah, after Katherina's talk, this seems very much like the sledgehammer approach to understanding and cycling of methylated compounds. But anyway, let's go for it. I think, you know, what the sort of processes that you've been studying and talking about might explain why some of these processes don't work perfectly and that sort of thing. So it's quite pretty interesting, and there is a sort of continuity there. So when I think of, you know, the turnover of particulate methylated sulfur compounds, I think of sort of 4 different processes or types of processes that might be going on. One of them is, you know, the internal cycling of DMSP or related compounds, and, you know, what's driving that. And that's really, you know, down to the function of these compounds in the cells. And we all know there's probably multiple functions, and, you know, that might impact how fast those compounds are being internally cycled. And then there's processes like, you know, cell lysis, so autolysis, autolytic processes that cause the release of dissolved organics, including dissolved organic sulfur compounds. That sort of DOS is there. And, you know, we all know that if you grow phytoplanktons in a batch culture up to a point where they become senescent, then the DMS concentration, if they're DMSP producers, then usually goes up substantially. And the same sort of thing probably is happening in, you know, blooms when they start running out of nutrients and that kind of thing in natural waters. And then, you know, there's been quite a few studies now that have shown that, you know, viral lysis is a major sort of mortality for many phytoplankton. And when that happens, when the viral lysis actually takes place, then there's a a massive release really of all the dissolved organics, or all the organics into the dissolved phase, from the cells. Is pretty. And then there's the bit really that I'll focus on most in this talk, which is then the grazing and consumption of phytoplankton, DMSP in particular. I won't talk about bacterial DMSP consumption, but in some ways it's kind of similar, and you can get at it in similar ways or understand it using similar methodologies to some extent. But yeah, that would be the main focus of this talk. So really, in many ways, it started with John Dacey and Stuart Wakeham, who used the pretty well-established approach of taking zooplankton up, picking zooplankton out of a water sample and adding them to a culture phytoplankton and seeing what happened. And in this case, they added this Labidocera to a gymnodinium culture, and they saw a pretty dramatic increase in DMS production. And they added them at pretty high concentrations to 30 to 40 copepods per liter, for instance. And so that was, one of the first indications that zooplankton grazing was an important part of the DMSP and DMS cycling processes. And they were able to extrapolate that information to show that if the zooplankton consumed, say, 3% of the DMSP containing phytoplankton each day, then they would be producing a pretty substantial amount of DMS in that process. And those sorts of approaches have been, taken into natural waters or nearly natural waters. So I think Maurice is on this call, but yeah, in the study that Maurice and co carried out, so this was a mesocosm experiment. They added Calanus to the mesocosms and or the mesocosm water anyway, and tracked the fate of EHUX or DMSP and DMS and that kind of thing. And they showed that what seemed to be happening was that the Calanus was actually grazing on microzooplankton that were grazing on DMSP, producing phytoplankton, and that was generating an enhanced DMS in the incubations. And then, similar work's been done with krill by Daly and DiTullio, so grazing on ice algae, and even, that I think John Dacey was involved. I think Hill et al. I think the paper is they've even done similar things with Menhaden fish, so planktivorous fish as well enhance DMS production when you add them to a natural system in excess to some extent. But really what I'm going to talk about most is microzooplankton grazers and their impact on DMSP and DMS. And part of the reason for that is that, as shown in this sort of meta-analysis by Calvé and Landry, that in much of the ocean anyway, if you look at this fourth column over, then the amount of, microzooplankton grazing, if you believe the results from dilution experiments, which this is a meta-analysis of, so it's a whole bunch of dilution experiments put together, then they're turning over, between 20 and 80% of the chlorophyll, the standing stocks of chlorophyll that are present in these natural waters. And it kind of varies in their meta-analysis. It kind of varies with latitude to some extent as well. It seems to be a bit lower in polar waters, but that might be, a bias of sampling time and sampling place and that kind of thing as well. And when we're talking about microzooplankton, then we're talking about, in general, these sorts of heterotrophic processes like this tunicate ciliate or this athecate dinoflagellate or armored dinoflagellates, but also possibly, copepod nuclei or anything like that that's less than 200 microns. Okay, so that's the reason why to focus on them. Some of the early work that's been done in this area, and really interesting work in a way, use the same sort of approaches as we use for the zooplankton. So adding, combining monocultures of heterotrophic and autotrophic protists. And in this case, the grazes this Oxyrus marina, which many of us know. So that's a sort of model organism in a way, and grazes pretty voraciously on ehucs or Dunaliella in cultures. And one of the really interesting things that's come out of some of this work is this idea that DMSP might be a deterrence. And Wolf and Steinker produced this really nice paper in Nature at one point where they were able to use what turned out to be a really convenient sort of model, which was that some ehucs have strains of ehucs vary in their DMSP lyase activity by a huge amount. So this is on the right, there's the lyase activity on the y-axis and the DMSP cell content of the different strains on the x-axis sort of thing. So the DMSP content's pretty similar between most of the strains, but the lyase activity varies considerably. And that they figured out that might actually be impacting the grazing rates of the Oxyrus. So when you get more DMSP lyase in the cells, you're getting more of that DMSP transformed to DMS and acrylate, and that acrylate might be reducing the pH of the digestive vacuoles in the Oxyrus and slowing them down from grazing on the ehucs. So there might be a deterrence effect happening there. And it was a really neat piece of work. But many of us have looked at this in different ways, and we haven't really been able to substantiate that in most cases. And so if you take this question into natural waters, how do you go about it? What ways can you actually approach that? And what you want to try and do, I guess, is try and alter the grazing pressure on the phytoplankton community or the DMSP producing community, and then figure out what the response to that change in grazing pressure is. And one way of doing that is to use Landry-Hazlitt's dilution approach. So this is a sort of schematic of it on the left here. So you're taking a water sample, filtering it through some of it through a 0.2 or a 0.45 filter, and then adding that in varying amounts to the original unfiltered water. And in doing that, you're actually separating the phytoplankton and the grazers. So you're decreasing the encounter rate as you add more filtrate. And that's kind of what's plotted on the right here, the fraction of whole water on the x-axis and the apparent growth rate. So you can measure the apparent growth rate as chlorophyll or actual cell counts, or we'll see in a minute as DMSP. And the intercepts your growth in the absence of grazing, and the slope of that regression is the grazing mortality, the specific grazing mortality rate. So if you do this sort of thing and you use DMSP as one of the proxies of the DMSP containing phytoplankton, then this is some work we did in the North Sea. As part of the DISCO study. So these are the same sort of dilution plots here on the left, and I'm showing chlorophyll, DMSP, and ehucs that we've counted on the flow cytometer as well. And so you can get a rate of production of DMSP, so rate of phytoplankton production, specific rate of production from that plot, for instance, and a rate of consumption of that DMSP. And, when you say, in general, these results are pretty robust. So the statistics, the regressions are pretty significant and statistically robust. So you can kind of start to believe the values to some extent. And one of the interesting things, you can do is compare the different rates for each of those sort of components of the phytoplankton, and that's what I've done here on the right. So I've compared top right sort of growth, specific growth rates of maybe the bulk community, so the chlorophyll versus DMSP or EHUX. And you can see in most cases, this is a Lagrangian study, so this was like a 2-week long study, Lagrangian study, where we traced an EHUX bloom in the northern North Sea. Most of the time, the bulk sort of phytoplankton was growing probably faster than the DMSP containing components of the phytoplankton or the EHUX population. And it also looked like, you know, at the bottom here, that they were also being grazed at a higher rate than the DMSP and EHUX populations as well. So there's, you know, there's a differential going on there, and that might sort of support that deterrence idea a little bit as well. But it's important to understand that, you know, not all DMSP, even in an EHUX bloom, is EHUX, and there's lots of other sort of particulate DMSP in there. And as Marty pointed out in a previous sort of set of talks, you know, quite often that's the black area in these pie charts. Quite often you can't really explain all the DMSP by just taking the abundance of the different phytoplankton and allocating them a DMSP content per cell, for instance, and trying to work out where all the DMSP is in the particulate form. So, you know, it's just measuring DMSP and trying to figure out what the production rate is a little bit of a blunt way of doing things. And even more blunt maybe is to try and understand, you know, what's happening to the dissolved components in those dilution experiments. So, you know, when you carry out these experiments, you have to filter a whole load of water for the filtrate, and that has consequences, you know, when you start releasing compounds from the particulate phase into the dissolved. So it's really pretty difficult to set these experiments up in a way that they'll work and you can make anything meaningful of them. You have to be really, really careful with the filtration. But here we've tried to, so this is work we did in the Iceland Basin. And the top plots are, you know, your typical dilution type plots. So for a nanophytoplankton population that we counted on flow cytometer. And then the bottom plots are what's happening to the DMS or the dissolved DMS-PD in those incubations. And what you want to notice on the left is that I've plotted the actual nanomolar per algal fraction. So it's normalized to the algal fraction in each of those dilutions. So and when I say algal fraction, I mean the fraction of whole water. So it's related to how much we've diluted each of those level, how much we've diluted each level of incubation. And you can see in each of these, you seem to get an increased DMS-PD and DMS production with reduced filtrate. So the more grazing that's going on, so to the right of the plots here, higher grazing pressure, you're getting more DMS-PD and DMS produced. And I can go into these a little bit more if people are interested. So we're not the only people who have done this work. So the CMO group have done similar work, and this is work from the Mediterranean. So again, typical sort of dilution plots at the top. I'm not too sure. I can't remember what the two symbols are there. But in this case, they plotted net DMS production and the proportion against the proportion of the DMSP grazed per day. So they've calculated that from the dilution experiments, and with increasing amounts of DMSP grazed, then you get increased DMS produced. And in this case, they get something like 4 nanomolar per day being produced if you take, you know, the two extremes there. Okay, so one of the other processes I talked about was viral infection, and there's been some work on that and how that impacts DMSP particulate. and the transformation to the dissolved phase. So this was one of the early studies that looked at this by Hill et al. When they took Micromonas pusilla and added Micromonas pusilla virus to it in an axenic culture. And you can see at the top here, there's a real dramatic drop in the particulate DMSP, and that's transformed into dissolved DMSP. So it's a really effective way of kind of transforming one to the other. And this is the control at the bottom. But how do you get at that in natural waters? Well, that's really, really difficult, I think. And most of the time, you just have to rely on sort of correlations between infection rates versus the amount of DMS being quantified in the natural environment. But what you could do potentially, if you were brave and had lots of time, would be to try and carry out this sort of extra dilution experiment that we introduced at one point, which was aimed at obtaining a viral mortality rate, really. So you carry out the same sort of dilution experiment, but do an extra filtration step where you're actually filtering out the viruses as well. So in this case, you're decreasing the encounter rate between the viruses and the phytoplankton as well. And if you take the difference between, you take your regression, do the same sort of regression statistics, and take the difference between the viral-free incubations and the grazer-free incubations, grazer plus virus, I should say, for the top one, then you get potentially a viral mortality rate, so you can attribute that to the viruses. And if you did the same sort of thing, instead of in this case sort of measuring or quantifying the number of cells, you actually quantified the DMSP, you might get a turnover rate of DMSP due to viral mortality. There's lots of issues with this approach, and I was kind of responsible for coming up with it. It's definitely not perfect. Recently, I've published an alternative sort of way of manipulating grazing pressure, and that's by adding surrogate prey to natural waters. So when I say surrogate prey, an alternative prey to the natural prey that's in the natural waters. So that could be, in this, polystyrene beads or a different phytoplankton, or I'll show you in a minute, starch grains or something like that people have used. But how it works is that if you add more and more surrogate prey to a natural sample, then the grazers start to become saturated by that surrogate prey, and that saturation, so it's called a Hollings type 2 response, really. That saturation reduces the grazing pressure on the natural prey and allows them to grow faster. So it's the same sort of idea as the dilution approach. So if you then measure the apparent growth of the natural prey, that in theory should increase to an asymptote, and that asymptote is your growth in the absence of grazing. So you're close to gross growth if grazing is the main mortality. And the difference between that and the net growth, so when you don't add any surrogate prey, is your grazing mortality rate. And this actually works pretty well in the natural waters that we've tried it in so far. So we did a seasonal study in the Gulf of Maine here and looked at the grazing rates and growth rates of pico eukaryotes and Synechococcus when we added 2-micron beads to these incubations. And so these are the same sort of plots I just showed you. And so, as I said, we did it over several days, over a sort of summer, and found pretty robust responses, I suppose. So these fits are statistically pretty robust, and you can get growth rates and mortality rates out of that. And what we haven't done is measure the DMSP part of this. So if you could do this, so here we've just counted cells, but if you measured DMSP, then you might be able to get at that DMSP production and turnover as well. And what's probably even better about that approach in our context is that because you're not filtering any water, you're just adding particles to a natural water. You're not really disturbing the DOC to the same extent anywhere near. So what you can do, I hope, is that actually quantify. So if you measure DMS or DMSP dissolved, you could start to unravel the rates of production of those compounds under different rates of grazing mortality, sort of thing. And we did some preliminary work at one point in Plymouth where we added this Cruomonas salina, which produces very little DMSP, to a natural water sample. And so the more of the Cruomonas we added, so up to 10-fold the natural abundance of the nanophytoplankton, the less DMS we ended up with after 24 hours. So it kind of supports our hypothesis of how this might work. And so that's something that might be worth exploring as a method in the future. And I'm not sure why I put this in, but this is my last slide. But you know, in that case, in those cases, we just added like 2-micron beads, right? And so we're looking at a very specific size range of phytoplankton and grazer in a way. But you could add, you know, a whole range of different bead sizes to try and cover the whole sort of spectrum of phytoplankton and grazer in your natural waters, as long as it doesn't disturb the growth rate of the phytoplankton that are there. And I knew a guy called Kai Kivi. I don't know if anyone else knew him. He's a Finnish bloke, really, really nice guy. But he used, and he didn't, when I met him, he didn't have much funding, but he was using wheat starch particles to track the uptake of these particles in ciliates and things. And he is able to show that different ciliates had different sort of size preferences for that they, you know, grazed upon. And it was some really neat sort of low cost, cool work. But there's another really cool thing about Kai Kivi. He created the first punk rock band in Finland called Kola Kesta, and you can still find them on the internet these days. But yeah, sadly he's passed away. But yeah, that's all I've got to say. That was about it. But yeah, sometimes there's just cheaper, you know, quite straightforward ways of getting at these things, as well as more and more sophisticated routes that we're taking these days. Okay. That was it.
Great. Thanks, Steve, for this very wide historical perspective you gave us about this topic. And we have time for a couple of quick questions, I guess, before we move to the breakout rooms. So please go ahead.
Yeah, Steve, were they microplastic beads then you're using in those experiments? Essentially,
yeah, they're polystyrene. Yeah.
Polystyrene, yeah. So just thinking about the practicalities of doing that type of experiment, then do you have to carefully dispose of those?
Yeah. So we put everything through a same filter that we filtered, same cycle filter that you use for a dilution experiment, and just cap them all before discarding. Yeah.
Cool. Yeah, because I had a master's student a few years ago who did a completely different type of approach. So she was looking at whether microplastics are more palatable to copepods if they're infused with DMSP. So yeah, and so she used larger microplastics or microplastic fibers to do those experiments. So we were basically just soaking those microplastics in solutions of DMS and DMSP, and then feeding them to copepods, and they did indeed prefer the tasty DMS infused ones. So just wondering, when you're using those microplastics in that situation, would there be any adsorption of VOCs or compounds onto the microplastics that might influence the grazing dynamics? I don't know, just a thought.
No idea. but
No.
something. They're meant to be fairly inert, but yeah, absolutely could be. And the longer your incubation, the more likely that might happen. Yeah.
Yeah. Cool. Thank you.